Now coming to the alignments, right? So now I've given you a brief idea of how, because you have so many sequences available, you can do comparisons. And I've also told you, because genes most commonly originate via duplication of the original copy, and also these genes are vertically inherited. So therefore, sequence similarity is an indication of functional similarity. Also, it is an indication of evolutionary relatedness, right? So that is the whole idea of alignments here. So here you are. This is your, again, the same slide. So you have a common ancestry. So globin gene initially was a single copy, then it made two copies. They diverged from each other, forming the alpha and beta. Then alpha further formed seven copies, beta further formed six or seven copies, and then they took up, and finally they end up in different chromosomes in the human. The alpha globin locus is on the chromosome 16, and the beta globin locus is on chromosome 11, right? And together they form what is your the hemoglobin, right? So alpha and beta copies together form the hemoglobin. So here you are. This is your early globin gene. This is a gene duplication event. You have the alpha copy, you have the beta copy, right? So alpha and beta are basically paralogs of each other. And if you look at the alpha of mouse and alpha of human, they are orthologs of each other. So by definition, homology can be of two types. You can have orthologs or paralogs. Orthologs are genes that are related by vertical descent, coming from a common ancestor and encode proteins of the same function in different species. By contrast, paralogs are homologs that have evolved by duplication and code for protein with similar but not identical function. So they may have similar function but may not have an identical function, right? So again, one thing that is important to notice that homology is either there or it is not there. Homology is not quantifiable. It is either there or it is not there. Similarity is something that you can quantify. 80% similar, 90% similar. The more they are similar, the more likely to be homologous. But you cannot say it is like 60% homologous, right? You are either brothers or you're not brothers, right? So homology is either there or it is not there. So that is something that you need to keep in mind. And then, of course, if we move on, so why do you align sequences? Early in the day of protein and gene sequence analysis, it was discovered that sequences from related proteins or genes are similar, right? So if you have proteins of similar functionality or similar structure, if you go back and refer back to their sequences, you would find there is similarity in the sense that one could align sequences so that many corresponding residues would match. So alignment means what? When you put them together on top of each other, you would be visibly able to see that many of the nucleotides are similar in nature. You may have to adjust a little bit for the substitutions or for the insertion deletions, but largely you will see that the sequences are very similar to each other. This discovery was very important. Strong similarity between two genes is a strong argument for their homology or common descent. So if there is a strong similarity, remember similarity is what is quantifiable. Homology is either there or it is not there. It is not a percentage-based thing. So strong similarity between two genes is a strong argument for their homology or common descent, right? Homology means that two or more sequences have a common ancestor. This is a statement about their evolutionary history. Similarity is simply a statement that the two sequences are common by some metric, right? They have very similar, they're closer to each other with respect to certain metric. So similarity simply means that two sequences are similar by some criterion. It does not refer to any historical process, just a comparison of sequences by some method. It is logically a weaker statement. So when you say the sequences are similar, it is not a very biologically significant statement. When you say sequences are homologous because of the extent of similarity that is there, that is the actual biologically, laden and biologically loaded statement that you're saying. But in common parlance, they can be used for one another. For example, however, in bioinformatics, these two terms are often confused and used interchangeably. The reason is probably that significant similarity is such a strong argument for homology. So this is something that you need to keep in mind. So here we have two sequences, and you can see you could basically, there are two types of alignments that you can do. One that is known as global alignment. For global alignment, the sequence should be roughly of the same length. Whatever two sequences are comparing, they should be roughly of the same length. For example, here, this is around 67 bases, this is 70 bases, and you could put them together on end to end and see what are the positions where they are exactly similar, or if you could adjust a little bit here or there, can they give you more residues that are exactly matching, right? So this is global alignment. Entire sequences are aligned using as many characters as possible up to both ends of each sequence. Sequences that are quite similar and approximately the same length are suitable for global alignment, right? So here you are. Then the other type of alignment is a local alignment. So if you see here, if you do this global alignment, you don't see much of a similarity between the sequences. But what happens is within the larger sequence, you may have shorter portions that may be exactly similar. For example, here you can look at this. So this light blue, and this light blue is the same sequence. This red and this red is the same sequence. This blue here is also the same sequence. This dark blue and this dark blue is also the same sequence. So this is what is local alignment, where you're looking at small stretches of similarity between two sequences. In this case, one sequence may be extremely long, for example, the entire human genome. The other sequence can be very small, for example, the GAPD8 gene. So we are taking a 3855 base pair sequence and we're comparing to the chimpanzee genome to find where in chimpanzee genome do we have the orthologous region in chimpanzee, right? So that is what you're looking at. This is an ortholog then. So this is your global and local alignments. Again, local alignments, you could have several types of algorithms. One that gives you the best fit, which is the highest score alignment. Other where you can get the best fit as also the other possible alignments that are there. So this can be done here. This is local alignment. What happens is like the same, a similar sequence I'm showing you here, 67 and 68 bases, for example, here. And you can see if you do not do anything to them, just look at what are the positions where they're similar. Then in that case, you can see that there is very little similarity between them, right? However, if you introduce a gap, for example, here, if you introduce a gap here, what you have, so what happens is when you're aligning sequences, sometimes you have to introduce some gaps, and gaps can give you a better picture of the biological alignment that is there. So here what has happened is very clearly there is a one nucleotide insertion in this sequence. So when you accommodate for that insertion here by giving a gap on the top sequence, then you see they align beautifully with each other, one-to-one correspondence for every nucleotide that is there. If you don't do that, then there is a problem. So this is not a perfect string matching exercise that you're doing. You may have to adjust a bit so that you get the best match possible, the optimal alignment possible. What is an optimal alignment? Optimal alignment will have maximum score, minimum mismatches, and most maximum matches, right? So this is something that we're trying to do as an effort in case of alignment of sequences, and there are several algorithms that can do the alignment for you. So to start with, we start with the very basic algorithm called as Dotplot algorithm, and I'll show you how that works. Then you have what is known as dynamic programming algorithm, which is also a way of scoring the matches and then finding out the best alignment. And then finally, we move on to what is known as BLAST or heuristic algorithm. So we'll talk of all three and we'll do the practical one, BLAST, right? So let me just take you there now. So here we are. So gaps, what do gaps represent? Gaps represent micro or macro evolutionary events exclusive to one of the two sequences being aligned, right? So these are the two sequences that are being aligned. In this one here, there has been an insertion. To accommodate for this insertion in the bottom sequence, you introduce a gap on the top sequence. When you do that, you get a perfect alignment of the remaining part of the sequence. So this is biologically more informative alignment because you have introduced a gap and you are now able to see very clearly that there is a indel in between. And this one is a biologically illogical alignment. This will not fetch you any information. This can only tell you, okay, there is some similarity at the start of the sequence and there is no similarity whatsoever. Some random similarity at some positions, but largely the sequences are dissimilar. So therefore, alignment is not a simple problem in biology. You have to basically accommodate for the biological phenomenon that best explains and still gives you the most matching residues when you are comparing the two sequences together. Everybody with me, class? You're following?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Good. So then we move on and we talk of optimal alignment. So what is an optimal alignment? The alignment that gives you maximum matches, minimum gaps, minimum mismatches, and highest score, right? And this is generally thought to be biologically most logical alignment that you could get, right? So what we're looking for when you do an alignment is not one-to-one, just putting the two sequences together and finding out which positions is the nucleotide matching. What we're looking for is an optimal alignment. Optimal alignment would be something that has maximum matches, minimum gaps, minimum mismatches, and highest score, right? And generally, this would be the biologically the most logical alignment that you could get, and more explainable in terms of the biology behind the event. So that is what is an optimal alignment. Now, if you look at the alignment algorithms, there are two types of algorithms that are there. One is what is known as non-heuristic. The other is known as heuristic. What is non-heuristic? Non-heuristic is very rigorous. It finds the best and exact solution to a problem. But because it is finding the best and exact solution to a problem, it is very slow, right? And for example, if you're querying a 300,000 sequence database with a 100 base per query, it will take more than 2 to 3 hours. The algorithms you use here is what is known as dynamic programming algorithm. Heuristics, on the other hand, is what is known as an intelligent guess, right? So heuristics is different. We talk of Smith-Waterman and Edelman's, which are non-heuristic alignment algorithms. BLAST is a heuristic algorithm. So what does BLAST do? Let's say I give a query that will first break the query into smaller pieces. It will first see whether the small piece has a good alignment or not. Once the small piece is aligned well on the particular search space, then it will extend it and see whether this can be extended beyond a point or not. So this is an intelligent guess. It gives you it may not give you the best solution, but it gives you the near optimal solution. The time that is taken is 50 to 100 times lower. So therefore, this the time versus sensitivity issue is there. But most often you get the best alignment and at a much quicker time as compared to what you get here. So therefore, heuristics-based alignment algorithms have been very common, and BLAST is one of the most cited papers that you can ever come across, right? So this is something that you keep in mind, that in terms of algorithms, BLAST is a word-based algorithm. It has a certain, it will break your query into certain small fragments. First, check whether this word has a perfect alignment on the search space or not. If in the database it has a perfect alignment, then these positions will be extended. If it doesn't have, then of course you exclude that region from your alignment trials, right? So therefore, progressively, you are rolling out many regions from the genome for an alignment trial. You don't have to do the full sequence to roll it out. So that saves a lot of time here. All right, so here we are. There are three basic algorithms. You have Dotplot, you have dynamic programming, you have word method, right? So Dotplot is basically you can put the sequences on your X and Y axis, the two sequences that we're comparing, and then put a dot wherever there is commonality. And if there is no commonality, you don't put a dot. So for example, T and T, you have a dot. For example, A and A, you have a dot, right? So what this does is if you get a central diagonal end to end, the two sequences are exactly similar. Otherwise, when you get something like this, so there is a part that is similar here, there is a part that is similar here, in between there is some insertion deletion or something that is happening. So diagonals basically reveal the diagonal lines reveal the sequence alignment. Interruptions in the middle of a diagonal line indicates insertion or deletion. Parallel diagonal lines within the matrix represent repetitive regions of the sequences. For example, here is a repetitive sequence, this one, right? So parallel diagonal lines can indicate your repetitiveness. So this is not used anymore, but initially, to begin with, dot plot was used, right? So dot plot advantages readily reveals the presence of insertion deletions and direct or inverted repeats that are more difficult to find by other methods and more automated methods. Disadvantage, most dot matrix computers program do not show an actual alignment, right? So they will only show you diagonals and you'll have to then pull out those corresponding coordinates and see. And this is non-quantitative. You are not scoring for any match or mismatch or gap or whatever, right? So therefore, that's the important part. It is non-quantitative, does not return a score to indicate how optimal a given alignment is. And an improvement of this is what is known as the dynamic programming algorithm. So dynamic programming algorithm basically breaks the complex problem into smaller problems and then moves on. So basically what it does is it again finds an optimal alignment when matching two sequences for all possible pairs of characters between the two sequences. So if you see here, I'll just show you the diagram here. So here is your, let's say you have two sequences here. You have, this is your pattern here, mismatch, match, and gap. So G and G, they match, so you give a score of 1, right? Likewise, and what you also do is you take into consideration what is the parallel scores in the remaining three blocks. So here the highest score is 0. To that you add +1, it becomes 1, right? Then likewise, if you move on, you can look at this block here. So again, you're looking at these three blocks on the top, and there is already 1 here. And then this is a match time in to time in. So you say 1 + 1, it becomes 2, right? So this basically is a method of scoring with respect to 1, what is the current position you're looking at, whether there is a match or not, and then also keeping in mind the previous scores that you have given. So this generates a matrix, and that allows you to generate a alignment that is most optimal, right? And this is what is known as the dynamic programming algorithm, or DPA. Again, because every nucleotide is compared to every other nucleotide here across the length of the sequence, this will going to take a lot of time. This is compared to what is known as, and you have your Needleman-Wundt and Smith-Waterman algorithms, both follow dynamic programming algorithm. One is for the global alignment, the other is for the local alignment. This is followed by what is known as BLAST, or which is a heuristic algorithm, right? Or which uses what is known as the word method, or what is known as K-tuple, right? So we come to BLAST now, and we talk of BLAST, and we do a practical on BLAST another 5 minutes. So it works by finding words, short stretches of identical or near identical letters in two sequences. So what does BLAST do? How is BLAST much quicker than any other of the previous algorithms? Because it is not searching the entire, let's say you've given 100 base pairs, it is not going to take that entire 100 base pair and see how well it is matching to a region by scoring and everything, and then say, okay, did not match. What it does is, of the 100 base pair, it will break it into smaller pieces. It will take a 24 base pair stretch, first match that. If there is a reasonable match, it will extend it. Otherwise, this region can be excluded from further comparisons. So progressively, a lot of the larger database where you're searching for gets excluded, and only a few regions where there is reasonable perfect match, you can take it forward. So this is, saves a lot of time, and that is where the strength of BLAST is. The BLAST is much quicker, several magnitudes of order, at least 100 or 200 times or 1,000 times quicker than the previous algorithm. So that makes it the tool of choice for local alignment. Remember, we are not doing global alignment here. We're doing local alignment, which means that the query can be very small, and the database in which you are searching can be extremely large, right? So we'll do an exercise where we'll find a GAP-DH homolog in ortholog in chimpanzee, and that would be basically 3855 base pairs of human GAP-DH gene blasted against or compared against the entire chimpanzee genome, which is again 3.2 billion base pairs. So that is an exercise we're going to do now, right? So this is quick. I've told you this is heuristic alignment algorithm. It is very quick, divides the query into short words, and it initially only looks for exact matches of these words, then tries extending the alignment. Much faster, but can miss on some alignments, which is fine. It is going to give you the near optimal, not the best alignment, but given that it gives you a lot of efficiency in terms of time, this becomes your the best option possible, right? And then, of course, you can have wherever you want to introduce a gap. For example, in the previous sequence, as I showed you, you would have also introduced a gap penalty, right? So gap penalty is what you introduce here. And then, of course, we can do the practical first, and maybe if time permits, we can come up here. So let me take you to the practical question here. Here is the question. Illustrate the similarity and differences between chimp GAPDH gene, between the GAPDH gene in human and in chimpanzee. This is the question. Specifically, what you want to see is mention the chimpanzee coordinates in the human and mention the genomic coordinates of GAPDH gene in chimpanzee genome. You have to pick it up from the human and find its ortholog in chimpanzee and mention the coordinates in chimpanzee. Also, you have to quantitate the extent of similarity that is there between the two sequences. And we're talking of nucleotide blast. We are going to only do one type of blast today. That is going to be nucleotide blast. And tomorrow you will have your phylogenetics. So there you could have other types of comparisons as well. So here we are. Let me take you to the to BLAST, right? So let me just take you there and we begin. So first thing is we need to retrieve the sequence of GAPDH, right? So for that, we have done it multiple times now. We'll go to UCSC genome browser, right? You press enter. You come up here to UCC genome browser, home, and then of course to genome browsers, and then you go to the Asia site or the other site if you want to, whichever way you want to do it. And then of course in the genome browser you could just say it is loading human, and GRCS38 you say sorry, GAPDH. Everyone with me, class? You're following?
Yes,
Yes,
sir.
sir.
So you say go, and you have your sequence now, right? And our question is, what is the orthologous locus in chimpanzee where the GAPDH gene is found in chimpanzee genome, right? So for that, we need the sequence. So we'll just take the gene sequence here. So we go to view, and we go to DNA, and we pull out the sequence, right? So we don't need extra flanking sequence. So this all you know already. So we'll not spend time on this. And then, of course, we don't need to do a reverse complement because gene is on the process strand. We don't really need to mask repeats here because we are, if at all you want to, you can do it to lowercase because we want the ortholog, so the sequence has to be visible. Only then it will be looking for its match in the chimpanzee. If you do a capital N and there are some repeat nucleotides, they would also become capital N, and then they would be blind to that, right? So therefore, in this case, you are not doing a hard masking. You only do a soft masking because you want the nucleotide to be visible to the software that is looking for the orthologous locus in chimpanzee, right? So we say get DNA, and here is our DNA, right? So this is our query 3855 basis of the GAPDH gene sequence in human, right? Then the question is, what is the location of this sequence in chimpanzee? Orthologous, identify the orthologous locus in chimpanzee. So we go to NCBI BLAST again. So we say NCBI BLAST. Here you. So BLAST, basic local alignment search tool. As the name indicates, it is a local alignment tool, right? It is only going to find a local alignment. It is not worried about whether your sequence has a complete match, end-to-end match with your query sequence. You have a small stretch of query that you want and that you want to see where it is in the chimpanzee genome. And local alignment makes a lot more sense in this case because there may be differences between human and chimpanzee genome, but genes are almost completely conserved, right? So we look at this. BLAST finds regions of similarity between biological sequences. The program compares nucleotide or protein sequences to a sequence database and calculates the statistical significance, right? So this is again the program compares nucleotide and protein sequences and calculates the statistical significance of matches. BLAST can be used to infer functional and evolutionary relationships between sequences as well as help identify members of gene families, right? So this is what you have. This is the current version of BLAST here. We have several flavors of BLAST. For example, here you have nucleotide BLAST, which is basically the input, the query sequence is a nucleotide, and the database where you're searching for match is also a nucleotide. Then you have likewise protein BLAST, which is called as BLAST P. And here the input sequence is a protein, and the database in which you're looking for match is also a protein sequence database. In between, you have something called as BLAST X, where you have a query that is a translated nucleotide, and you're looking into a protein database. And then you have a T-BLASTn, where you have a protein as your query, and you're looking at a translated nucleotide as your database. So this is not something that we use commonly, except that when you have something like this. But what we most commonly use is either BLAST P or BLAST N, right? And if you talk of DNA, it has to be BLASTn. So what we'll do also is, because we know that we want to search only in chimpanzee, so we'll select our genome here, right? Common name. So I'm basically putting up CHIMP, and now if you see, it is going to prompt me. So I select for chimpanzee troglodytes, which is the pan troglodytes or the chimpanzee ID, right? And if you see, the ID is the same, 9598 or 9598 here. So this is your the normal chimpanzee, which is called pan troglodytes or chimpanzee troglodytes. So we'll select this, right? So we have selected our genome into which you're going to do a nucleotide BLAST. So we say search. So now we have defined that our database is going to be a genome of pan-tro3 or pan troglodytes 3, which is the chimpanzee genome, right? Then here you can paste your sequence. So we are on BLASTn. You can see by default it has taken us to BLASTn because the database is a nucleotide that we selected. Nucleotide database is selected. Now we copy our sequence here. Anybody with me, class? You're following?
Yes, sir.
Okay. So here we paste our sequence in the FASTA format. You can directly copy from there and paste our sequence here, right? And then, of course, if you want to, look for options, you can try and look for algorithm parameters, but generally we go by default. But I'll show you what parameters are available. Before that, here, so we are optimizing for highly similar sequences, which means the initial word that we take will be larger in size. So we take a word size of 24. Normally the word size is around 7 or 11, right? So we're taking 24 as the initial seed. Only when there is a perfect match to the seed. Remember, it is a word method or a k-tuple method. So first, it will find a match to your word. If there is a perfect match to the word, only then the search space will be extended to 5' and 3' ends of the initial 28, 20, 24, 28 base sequence that you've taken, right? So this is what is known as mega blast, where the word size is larger. You could do a normal blast where the word size is smaller. The smaller word size means that you are giving lesser astringency for the initial match. Larger word size means you are giving a very strong astringency for the initial match. So here you are, you say program selection, you're looking for highly similar sequences, which is mega blast, right? If you want to look for somewhat similar sequences, that will be a normal BLASTn, right? Then you could click on this blast button to see what is it. But let's first look at the algorithms parameter, right? So here you are, if you see maximum target sequences is 100. Then you have expected threshold value, E value. We'll talk of this as we go along. A minimum of 0.05 is what you want. Word size, if you see, is 28. So this is mega blast, right? And then, of course, you have your match and mismatch score. So 1 and 2 is what you're giving as your match and mismatch score. Then there's also a gap cost that you're giving. And then, of course, you are filtering for repeats and for low complexity regions. So this is what is your options that you have. We are doing mega blast because you've selected a size of 28. If you see, there are options for 16, 20, 24, 28, right? So we're doing mega blast. We're looking for mostly similar sequences or highly similar sequences. Then we say blast, right? So this is the button that will take you to submission of your sequence for comparison with chimpanzee genome. So here we are. If you see, this is the results page. First on top, there is a job title. So this has been taken from your def line of the FASTA sequence, right? AG38 DNA, and then you have the locus and the 5' pad 0 and so on and so forth, which is basically part of your def line. Here, if you see, this is the def line of the FASTA sequence, this one. So it has taken this one as its ID or, the job ID or job title. Then this assigns a random ID for this, right? Then, of course, the program you're using is BLASTn or nucleotide blast. Then the database where you're searching is your pan-pro3, pan-troglodytes 3, assembly number 3, right? This is what it is, right? Then you have query ID, which is also generated here. Description is the same as what you've given in your def line again. Molecule type is DNA. You didn't specify at any point of time that you're giving a DNA, but it identifies it as DNA. Query length is 3855 bases, right? Then if you go down here, the results come in 4 tabs. First is the description, where all has it found the match, right? Then you have a graphic summary, which shows you that one of the matches is absolutely complete, the others are partial, right? Then you have the alignments, where you'll have the alignment, we'll come to it, and then you have the taxonomy, where did it find the match? So since you have only selected for chimpanzee genome, so all matches are to the chimpanzee genome. Whatever hits are there, they're on the pan-troglodytes genome, which is a primate genome. So and the total number of hits you've got is 22. So coming back to the description page here, if you see, you will have your statistics for the match. For example, here, there's something called query coverage. So what fraction of your query found a match here? That is important. So if you see here, there is one match at 100%, which is a perfect match. I mean, the entire query has been found as a match at a single location. Others are very small. If you see, this is 33, 31, 13, 23, and so on and so forth. So these are just matches by chance, probably, and only to a fraction of your sequence, not to the complete sequence, right? So query coverage is 100% only in the one of the heads, and which is our major head, right? E value is the expect value. What is the random chance of this match being found by chance? So there is no chance here. It is 0% chances. This is a true match, you can say. The closer the E value is to 0, the more confident you are of your homology, right? The more confident you are of your match being found, because there is a certain level of relatedness between the sequences. Then percent identity is basically how many times A matches to A in the alignment, T matches to T in the alignment, G matches to G, and C matches to C. And you can see there is high percent identity, 98% identity. For others with the smaller query coverage, there is still high percent identity, but that is only to a part of your sequence, which means that this may not be a very significant match, right? Then you have the accession and everything, and the accession number, right? So this is what is your major statistics that you have. And if you see here, there is only one match that is to 100% query coverage. Remaining matches are small in size, and therefore by virtue of being small in size, they may not be so significant for our identifying the orthologous locus in chimpanzee for the full sequence, right? Then if you see here, yes.
Sir, could you just explain the E value again, what it is?
So E value is the expect value. It is basically a value which indicates that your match that you've got is not a random by chance match. It is a true match, right? So 0.0 means that it is a true match, and it is reflecting homology. You could not have had this match unless the two sequences are related to each other, right? So that is the whole idea. We'll talk of it more as we go along. So E value or expect value, right? The lower this value, the better it is. Ideally, when you're taking any hit as your significant hit, the E value must be 0.0, right? And or the lowest possible value is more significant. Like 1E173 is not so significant, right? Although the query coverage is 31% and this is 93.5 or 0.32% of percent identity. So this one is the best hit here, undoubtedly. So we can take that. We look at the graphical summary. We have already looked at the alignments. So here is our alignment, right? And if you can see, this is the complete alignment here. This is the larger alignment, and then you have this small fraction alignment, the 31 base pair alignment that you have. This is coming up here, right? So let's look at the best alignment here. So if you go down here, this is your alignment, right? How does the alignment come? Your query will be on the top. Your query would start with nucleotide 1, and until the whatever portion of the query coverage has been matched. So for example, here, this is the full alignment. So you have until 3855. Then you have partial alignment. For example, here, in your query, the position that matches at the start is 3067 until 3485. So you can see this is only just a partial alignment. So this may not be the correct alignment. And this is definitely not the orthologous locus you're looking for, because you have a better hit here. So you take that as your consideration. Everybody with me, class? You follow?
Yes, sir.
Right? So,
Yes, sir.
Good.
Yes, sir.
Okay, males, you're following? I don't hear any boys speaking for a male.
Yes, sir.
Okay, good. So here you are. So if you look at the top here in the results, we are looking at the alignments, right? First thing it says is the database to which you're looking at. This is pan troglodytes. This is the length of the sequence that you're looking at, right? And then of course you're looking at chromosome 12. So this is the length of the chromosome 12 in chimpanzee, right? So the match is found on chromosome 12 in chimpanzee. This is 138751763 bases long, right? The number of matches found are 6 here on this chromosome. And then of course the main match is here, right? Then if you look up here, where does our sequence match? The 123855 base pairs match to in chimpanzee from position 1236722, which is the starting coordinate here, if you see also, until the position, this one, 123711591. This is also mentioned on the top, if you see, right? So I'm just showing you that if you look at the alignment, how does it look? But let's get to the top. So this is what it is. This is 123677222 123711591 on chromosome 12 in chimpanzee, pan-troglodytes, right? This is one thing. Second, the E value is 0.0, which means this is not a random match. It is a match because there is certain level of relatedness. Then you have identity. How many of the nucleotides are exactly similar? Of the 3873 to which it is matched in the chimpanzee genome, 3807 nucleotides are exactly similar, right? So 3807 of the total 3873, the orthologous locus length, 3807 nucleotides are exactly similar, which translates to 98% similarity, right? Which means that at that position there will be mismatches, or at positions there will be some gaps, right? And we'll look at those. How many gaps are there? There are 21 gaps in the alignment, right? And the matching has happened to the plus strand on both the DNA. So we had isolated plus strand, and on the chimpanzee also, the genome, it matches to the plus strand only. So now if you look at the alignment here, class, everybody, if you see here, this one, there is a cytosine here, there is a thymine here, right? Can you see that on the alignment?
Yes, sir.
Wherever there is an identical line, identical match, you have this vertical line. So the first position where it is missing is somewhere here. You can see you have a cytosine on the query and you have a thymine on the subject, right? On the subject, yes. So in this case, what this is a substitution, right? So you have a thymine here and a cytosine there, right? Then again, if you see here, you have a guanine here, you have an adenine here, right? So this is also a substitution, right? The nucleotide has changed in human or in chimpanzee. This you cannot say, but there has been a change in the nucleotide at that particular position. Then if you move down, you have an A here, you have a G here. This is also a substitution. And then if you further move down, you will see a gap, right? Let us see a gap. For example, here. So this one here, 901, can you see? There is a huge gap that is there. So there is a extra sequence in the chimpanzee genome, which is not present in the human genome. Remember, our query is from the human genome, right? Everyone,
Yes, sir.
query is
Sir,
from the human.
so please explain the, it means small letter alphabets in the above row.
Those small letters basically are your low complexity region. So you'll see there is a whole lot of GGDDD coming in. Can you see that? So this is
Yes, sir.
basically low complexity region, right? It may not have too much of information. So therefore it is showing as small letters. There is no
Okay.
other thing to it, right? So then you move on, and if you talk of this, so we were here at this position here, 901, and you can see there is a whole lot of sequence missing in the human, which is our query, and there is some extra sequence present here in chimpanzee, right? And then of course, from beyond that, the alignment is again perfect. So these are some of the things. What you'll see in alignment is one, mismatches, which will be marked with absence of this vertical line. There would be a say, different nucleotide here and another different nucleotide here. These are mismatches because these are substitutions because the nucleotide has changed, but otherwise the next few nucleotides are exactly the same. Then what you'll also have is something like a gap here. It could be a small gap, for example, one nucleotide here, or it could be a long gap, right? So which is indicative of the indels or insertion deletions that are happening during the course of evolution that you see. And then of course, largely you can find the orthologous locus here in chimpanzee, and you can say the human GAPDH gene is represented at locus 1, 2. Again, you can go back to the top and pick up here. So for example, here you could pick up this one. You could say that the GAPDH gene, the orthologous locus for GAPDH gene in chimpanzee is 1, 2, 3, 6, 7, 7, 2, 2, 2, 1, 2, 3, 7, 1, 5, 9, 1, right? And this was the similarity level. So if you remember the question we had asked, let me take you to the question again. What I told you was, one, illustrate the similarity and differences between the GAPDH gene in human and chimpanzee. To look at the similarity and differences, you look at the alignment and also the identity score. And then, of course, you're looking at the genomic coordinates in human and in chimpanzee genomes. Genomic coordinate in human you already have from the UCC genome browser. Now you know the orthologous locus genomic coordinates also. And then, of course, you're quantitating the extent of similarity by the similarity score. The identity or similarity is same in case of nucleotide sequences. So when you do a BLASTn, identity and similarity is the same, right? Because If A matches for A, T matches for T, G matches for G, and C matches for C, or not at all. When you're doing the same for protein sequences, the identity and similarity are different, right? So you may have a different amino acid which is of the similar class of the amino acid that was present earlier in the other genome. So therefore, identity is different from similarity in case of protein sequences. It is exactly the same in case of nucleic acid sequences. You follow class, everyone?
Sir, I have a query. Can I ask?
Yes, sir.
Yes, please do.
Sir, in that gap, what will we say? Means, how can we interpret this as it is means the insertion in the chimpanzee or the deletion in the human genes?
So that you cannot say unless you compare many other such genomes and then find that, okay, in chimpanzee it is always present and in human it is always absent, right? It could be your reference genome ascertainment bias also, It could be
Okay.
that, so basically what we prefer to call it as indel. You don't call it either insertion or a deletion. You say it is an indel. It could be an insertion in one and deletion in the other, or vice versa.
Okay, sir.
You get my point? So therefore this term indel has come, right? So
Okay, sir.
if you have multiple sequences and everywhere in chimpanzee you have it and you do not have it in human, then of course you may say that it is a post divergence deletion in human and it was present in chimpanzee earlier. You get my point?
Yes, sir. Yes, sir.
So
Thank
those
you, sir.
are slightly difficult to infer. Depending on what data you have, you would be able to infer that, right? But here it is clearly a indel. It is not present in the human genome. It is present in the chimpanzee reference genome, right? That much we can say for sure. Whether it came in chimpanzee after the humans diverged or whether it was lost in humans after divergence is something that you cannot say from this alignment. We're looking at homology. We're looking at orthologous locus in chimpanzee. One last thing that I need to tell you is here on top, you could also do a filtering, right? For example, you could filter for percent identity, or you could filter for query coverage, right? Or you could also filter for e-value, right? So e-value, you could give a range that I do not want anything below this. That will filter out. Percent identity, you could say I do not want anything below, let's say, 95% percent identity. That will filter out. Query coverage also. So that would depend on your results, right? So if you look at the descriptions, you would know which would give you the filtering power. So for example, here query coverage is the best filter for us because when you say query coverage and you say more than 90, only one hit will come up, nothing else will come up. If you talk in terms of percent identity, if you say 98% and more, then again only one is going to come up, the rest of them will be lost. This is also 94%, but the query coverage is less. So you could use both also, you could say for percent identity, you could say, okay, 90, let's say we say 85 to, let's say 90, let's say 100, right? Okay. And then for query coverage, you say 80 to 100 again, right? So now you're using two fields for filtering and you click on filter. This will filter out everything. It will leave you with ideally with just one hit because that is the one that satisfies both the conditions, right? So percent identity is greater than it in the range of 85 to 100, right? This is the percent identity. And then the query coverage is greater than 80%. So only one hit remains. So you could filter because you'll get a lot of nonsense. It's also, as you can see, there's a small thread that finds multiple matches elsewhere. You could have, let's say, a repeat sequence in your gene sequence also in intron. So that repeat sequence will go to multiple places in the genome and give you hits. So therefore, those filtrations are important. Two important filtrations are one with respect to e-value. The other is also with respect to query coverage, right? The query coverage also has to be significant. If you only find a 10% of your query matching at 100% identity, that is not a good match for you, right? So therefore, these are some of the criteria that you can use to filter out from a long list of matches that you get, and then finally get your final inference from the sequence. So that is BLAST for you. There is a little bit of difference in protein BLAST because protein BLAST, again, as I told you, the identity is different from similarity, and therefore the scoring is slightly different there. You have to basically also consider if there is a substitution, what is the new amino acid, whether it is similar to the previous amino acid in terms of its chemical functionality or not. accordingly, again, you'll have to do your scoring scheme a little different, right? And that is where you have your PAM matrices, point accepted mutations, that come in. So these are pre-calculated scoring matrices that you can use for your understanding. So I think we can close here. It is already around 1.5 hours of lecturing. If you have questions, you can ask me. Otherwise, we'll close here. Other questions I can give in the group if you want, right? So somebody has mentioned that she has a transcript but ID, but is not able to basically get the genomic coordinate. Ideally, that should not happen. But if you pass on the transcript ID to me, I can have a look and let you know, because that transcript ID will be somewhere in some genome, right? It is coming from some accession number, so it should ideally be able to take you to the original genomic context also. Otherwise, you can go back to the paper from where you're picking up this transcript ID. That would definitely have the genomic context as well, right? So it should not be a problem, but it requires a bit of a search. Sometimes it so happens, and it is very common, that when you need a very small piece of information, which may be very specific to you, you'll find NCBI is so full of information that it is like finding that needle in a haystack. It will be there in NCBI or in UCC Genome Browser, but to get to that becomes difficult. So that is the problem of having a very exhaustive database that you have, right? Question?
Sir, how much percent similarity is required for us to say that two sequences are homologous?
So that is also an important question. So if it is a DNA sequence, it should be more than 80-85%, right? For protein, this zone is around 35%. Because proteins can have a lot of variability in terms of having a similar amino acid and not the identical amino acid. But for DNA sequence, identity and similarity are same. So therefore, DNA 80-85% similarity, identity, and the query coverage again becomes important, right? The entire query, if you're looking for the orthologous locus, it cannot be just 10% of the query is matching with a high percent identity and you take it as good. So for nucleotide sequence, the percent identity must be 85% or more, right? In some cases, there could be a bit of borderline case, but then generally 85% or more is taken to be a good inference of homology, right? If it is not so, then of course they're not homologous. For proteins, this is quite low. And in fact, if you go through some of the basic chapters on protein alignments, you would have this graph that defines, okay, that 5% or more, if it is identity or similarity, then in that case, this could still be considered a homologous protein. That is also because, proteins, the entire sequence is not the functional part of it. You will have domains, you will have motifs, and then you will have some active site where several different amino acids far apart from each other would come in a closed three-dimensional space. So for proteins, this is slightly different. Very good question, I must say, right? But I hope I have answered you
Yes.
to your satisfaction, right?
Yes.
Yeah. Yes. So if there are no more questions, we can close now. It has been a really wonderful 4 days with you all. I have tried to be as exhaustive as possible because I wanted to give you not just the practical information, but also the base information that comes along with it, right? And again, most of these lectures are there on my YouTube channel. Anytime you want to go back there. And more so, again, I'm emphasizing if you are students or faculties, maybe if you are in a permanent job, you may not be so much under pressure. But if you're students and beginning your career, it would be nice if you could get into programming. Either R or Python is fine, but one of the two languages you should know, because the field is strongly moving towards it is biotechnology is more becoming a data analytics science. You have a high throughput data generation factory that generates a lot of data. It cannot be manually analyzed. It has to be done programmatically. So the sooner you get into this programming business, the more better you would be for finding a good postdoc. I went for a postdoc 10 years after my PhD. And the only reason I could get a postdoc 10 years after my PhD, not doing too much of science in between, because I was mostly teaching at MIT and then at Chandigarh University, was because I knew coding. So coding can give you that additional advantage that not everybody has as of today. So you could give a tryout. You could try my lectures. You could try somebody else's lectures. Give yourself some time also. It is not that on the first day itself you'll start learning. It will take some time. There is some logic to it. It's like a new language. Any language takes some time to pick up, right? So therefore, give yourself a fair chance. And if you get to know it and if you start enjoying it, you would have an advantage over anybody else who is competing for the same position. So on that note, I'll close. All the very best. Take care. Right.
Thank you so much, sir. It was really, really very nice with you for this whole 4 days. And I agree with sir that coding is really, really important, particularly for computational biology and bioinformatics. And hope the students actually have been in touch with you. Please be in touch with sir so that we can learn more and more from him. And thank you once again, sir.
You're most welcome, Dr. Amita. We go back very long time.
Yes, sir, that was true. And I remember sir was with me debugging my code till 1 a.m. in the midnight at CCMB. I still remember that fine day when I did some silly mistake and sir was trying to help me out. Yes, sir. Thank you so much, sir. Yes.
Yeah.